Thank you all for tuning in today. So I'll be talking for the next 30 minutes or so about the work we've been doing in Biosharing, biosharing.org, where we're mapping data standards, databases, and data policies in the life sciences. My name's Peter McQuilton. There's my Twitter. I don't know, if I mouse over, can you see my cursor or not?
Yes, I can see.
Can you see the cursor there? Okay, great. Okay, so there's my Twitter handle. There's a Twitter handle for Biosharing if you're into that kind of thing. Now let's see if I can go to the next slide. Okay, so I'm going to start at the end, really, a slide that's normally found at the end of presentations. This is our acknowledgement slide. This is just to remind you that this is not my work. It's actually the work of a lot of people over a number of years, principally the work of Susana Santasansone, our principal investigator, and Philippe and Alejandra, along with Eamon Maguire, our web developer, who's designed a lot of the Biosharing website, and then our developers, Milo and Massey, and then the knowledge engineers, myself, Alison, Delphine, and Melanie. And we're all based over at the Oxford Research Centre at the University of Oxford. Okay. And I'll give you a little bit more background before we get into the meat of the talk. So a little bit of a timeline on Biosharing to really give you an understanding of how it's grown over the years. So it all began back in 2008 as the MIBI portal, as a way to harmonise the many minimum information guidelines that were coming out at the time, to put them all in one place so that everyone knew what was already available. In 2009, this expanded into the Biosharing blog, which served as a focal point for the community on not only minimum reporting standards, but also other data sharing standards, such as those from genomics, as you can see from this paper here. Then in 2011, there was a transformation really of the blog into a registry composing of standards, databases, and data policies. And since then, really, this registry has grown and evolved, both in terms of the depth of coverage of our content as well as the breadth of coverage across various different standards. During the time since 2011, we've raised funds. We've also done a great deal of community engagement, not only within ELIXIR, but also attending various conferences and workshops, and through the creation of a joint Research Data Alliance and Force 11 working group, which I'll be talking to you about a little bit later on, and also composing and building an international advisory board who keep us on the straight and narrow. Now in 2011, we of course have links within ELIXIR, and we're embedded in and work with a number of NIH BD2K projects. If you want to know a little bit more in detail about our timeline and how we've grown over the years, there's the website link at the bottom of the slide here, biosharing.org/timeline, where you can find out more. So, what is Biosharing? Well, it's a web-based, manually curated searchable portal that monitors the development and evolution of data standards, also their use in databases or data repositories, and the adoption of both of these in data policies from journals or funders, for example. And our aim really is to inform and educate a variety of different user groups, which I'll go on to talk about later. So we're trying to map a complex and constantly evolving landscape, and you can see that here in this diagram that I'm going to take you through. In the middle here we have the content standards, and these descriptors are essential really for the interpretation, verification and sharing of data sets. Often they're seen as the kind of invisible guiding hand between the flow of data. For Biosharing, we've split them into three to make them easier to understand. We have formats, terminologies, and guidelines. So in our formats, we have essentially conceptual models or schema and exchange formats for the data. A great example of one of these would be the FASTA or FASTA exchange format for protein and nucleotide sequencing, from all the way back, I think in 1983 when that was first developed. Then in the center there we have the terminologies, which you may know as controlled vocabularies or taxonomies, or thesauri, or as I prefer to call them, ontologies. These are structured hierarchies of well-defined terms that one can use to annotate an entity. A good example, probably the most successful example of an ontology, are the gene ontologies, covering biological process, cellular component, and molecular function of gene products. And then finally, we have the reporting guidelines. These are minimum reporting checklists, the minimum amount of information one would need to record about an experimental data set in order to place it into context to allow it to be reproducible, findable, and all the rest. A great example of a reporting guideline would be the ARRIVE guidelines for in vivo animal testing. So, where do these content standards come from? Well, some are created by standard creating organizations, which I'm highlighting here on the left. These are organizations that come together to design and implement standards for a particular area or domain. An example would be the CDISC Consortium for Clinical Standards, Standards in Clinical Research. On the right-hand side, a great many standards are created by grassroots community groups, people coming together, often in academia, to generate standards for their discipline. An example would be the Biodiversity Information Standards from the TADWIG group, or the Combined Group for Computational Biology Standards. So, a little bit more about the numbers of standards that we're talking about, if I can. I'll talk about that later, obviously, wrong side. So one of the things which we do is we don't just capture information about the standards in isolation, but we also capture the relationships between them, because standards often stipulate the use of other standards. A reporting guideline may stipulate the use of a particular terminology when recording information about a data set. So we capture the interplay between the different standards. And of course, these standards are used in databases and often created by databases themselves. So we capture which databases use which standards. And this is really useful if researchers are thinking of depositing their data in a particular database, because at the start of their work, they can think, okay, I want to deposit in this database. What standards does that database use? I will therefore use those standards in my work. So we capture that information, which can be very useful. And then finally, to complete the triangle, we capture which data policies recommend or endorse the use of particular databases or data standards. And of course, if they endorse the use of a particular database, implicitly they're endorsing the use of the data standards used by that database. So by capturing this triangle, if you like, of relationships, we can fully explore and describe and explore the relationship between data standards, databases, and data policies. So that's what we're trying to do. That's mapping the landscape. Now I'll talk a little bit about the numbers. So here you can see we've got the content standards, formats, terminologies, and guidelines. Some examples down here at the bottom, and then more importantly, the numbers involved. So you can see in biosharing, we have over 225 models and formats. And then in terms of terminologies, using those from BioPortal, you can see there's over 500 terminologies now available. We have a smaller subset of those that are hand curated in biosharing. And then our reporting guidelines, we have now 115 of those in biosharing. So it's useful to know that all these standards relate to each other, but it's actually a very complex thing that we're trying to map, because not all databases use standards in the same way, and you can't assume or presume the use of one standard will imply the use of another. So I'm going to show you an example that proves this point. On the left-hand side here, you can see two very well-known databases, GEO, Gene Expression Omnibus Database, and the ArrayExpress database, both covering gene expression. They both use the Miami reporting guideline, which is great. So you may think, okay, then if you use one standard, they're going to use very similar standards throughout for their very similar data. Actually, no, that's not the case. So GEO uses the soft format and the Miami minimal format to record their data, whereas ArrayExpress did use MageML, and now that's been deprecated, uses the MageTab format. And so, okay, they're now using different formats, perhaps they use the same terminology. Well, no, because use of the MageTab format stipulates the use of the EFO ontology. So you can see these two very similar databases actually use very different standards. So it's important to accurately map down to a fine detail the types of standards that are used in database. I'm now going to give you a brief advert, if you like, for a webinar which will be coming along shortly along in the ELIXIR series. And that's the use of biosharing in the CEDAR project. So the CEDAR project is aiming to serve machine-readable content metadata standards to generate templates, which essentially will remove the need for the standards, but will actually use them and, importantly, capture the provenance of the elements from that standard, from those standards in other work. So if I can explain that, on the left-hand side here, the high-level information about the metadata standards is all recorded in biosharing. And then as part of the CEDAR project, we're splitting these down into their elements, if you like, and providing the provenance back to each standard in biosharing. So you can see here, we've got a number of different elements with their provenance. And if you want to know more about that work, Mark Musen will be talking about this in a forthcoming ELIXIR webinar, I believe on the 21st of June. Okay, so now back to biosharing. I was talking about how we're mapping this landscape and how it's very complex. Well, one of the things that makes it very complex is that it's constantly evolving. And one way that we capture that information is through the use of these status indicators. So here you can see we have 4 status indicators in biosharing. The one which is most commonly found is this R, ready for use status. So this is for all those resources, be they a standard or a database, that is fully maintained, fully supported, has been implemented or used in various contexts, and is adopted by the community. So the vast majority of the standards and databases in biosharing have this indicator. Underneath that, we have the in development status. Now, this can be used if someone is building a database or perhaps creating a standard and wants to gain greater visibility for their work. So people know that they're working in that area, they can perhaps contact them for help or give advice. And so it's a way of promoting and raising the visibility of a new product. So we have an in development status there. And then we have, for those resources, in often cases many standards that have been deprecated as they're no longer supported or maintained, and in fact they've been subsumed or superseded by another standard. So they have a deprecated flag. And where that's the case, we work with the people who have made those standards to advise our users on what standard they should use instead, and we provide that information on the record. So this record has been deprecated, perhaps you should consider using this record instead. So we have those 3 indicators, and then the 4th indicator is status uncertain. This is where perhaps the website for a standard is no longer available, we've contacted the people involved, and we've had no response. So we're not entirely sure whether a standard is still being actively maintained or used, and so we essentially put this status there as a buyer beware. This standard may be useful, but may not be the best standard you could use. And it also acts as a cry for help to our community, because someone out there may know what's going on with that standard, and so they can get in touch with us and let us know. And just to say, like all the data in biosharing, these indicators are all manually curated by our biosharing curators and verified by the people who work on those resources. So you can be assured that this data is as accurate as possible. Why are we doing all this? Well, we're trying to help people make the right decision in terms of where to deposit their data and what standards they should be using. And on this slide, I have 4 different types of user, if you like. So starting in the top left, we have our researchers on the bench or out in the field, who will have many questions. I've picked out one out here, such as my funder's data policy recommends the use of established standards, but which ones are established, which ones are widely endorsed by funders, for example, and which ones are applicable to my particular data set. So you can answer that question using biosharing. And then we have down the bottom left-hand side here, the curators and developers, people working on databases or standards. And often you need to know what else is out there. Are you using the most up-to-date version of a particular ontology in your database? Are you using the most appropriate ontology? And you can use biosharing to search through and keep abreast of more recent developments in ontology development. In the top right, we have funders and journal editors, the people who generate data policies. Often they'll be generating data policies for areas that perhaps they're not very familiar with, and they can use biosharing to understand which are the mature standards out there, which ones have been adopted by the community and are implemented in many databases. So they can use biosharing as a way, in a sense, of judging which standards and databases to endorse. And then finally, we have librarians and data managers who all too often are approached by people going, I've found this data down the back of the sofa, it's 10 years old, it's in a wrong format, I don't know what to do with it, where should I put it, and in what format should it be? And so those people can come to biosharing to find that out. Okay, if you were to go to biosharing.org, this is our homepage, this is what you would see. So I'm not really going to go through a lot of this other than to say at the top here there's various ways to get to different cuts of the data. In the top right hand side here we have this educational section which can help you if you're not too familiar with standards or with how to use biosharing. We have over 40 frequently asked questions in there. And then in the center of the screen in the dark blue we have our search bar. On the left hand side here you can see our simple solar search. This searches all the free text across all the records in biosharing, and so if you're wanting to get a quick overview of what's available for a particular area, domain or discipline, you can type it in there and away you go. If you want a more precise search that perhaps produces fewer false positives, you can use our advanced search, which allows you to do some field by field searching and also benefits from our autocomplete, so you can get a more precise hit list from that. If you're not entirely sure what you want to search or how to actually search, you can use our search wizard, which is still quite embryonic, but please do have a look at that, and you can search by species and then domain to search through and refine your search list. And if you have any ideas for how we could improve that, please do get in touch and let us know, as we're trying to make it as useful as possible. What I wanted to highlight was really down at the bottom here, you can see our 3 different registries and you can see the numbers involved. So in biosharing at the moment we have just under 700 data standards, over 800 databases and over 90 data policies. So, and all of these are expanding all the time. Okay, so now I'm going to take you through one of our records. This is a standard record, but there's a lot of things on this record which are common across all of our records. At the top of the record, obviously, there's the name of the record, the status of the resource, so in this case, this standard is actively in use and maintained, and then an abbreviation for the resource, as often resource is known by the abbreviation rather than the full name. Just underneath that, we have a general information section, which is common to all of our resources, be it a standard database or policy. Here we have some free text prose that's been curated and checked by the resource, explaining concisely what the resource is about. Underneath there we have the home page, where the resource was developed and when, and then we have these tags here that tell you what species that resource is appropriate for and what particular data types or domains that entity is appropriate for. And clicking on any of these will produce a list of all the other standards and databases that are used in that domain. So that's quite a useful way if you want to explore what's out there. Just underneath there we have a selection of icons and text here that show you where this standard has been included in a collection or recommendation. I'll go on to talk about these a little bit later, but what these are essentially are ways of grouping together various records for a project, organization, or data policy. Underneath here you can see we have when a resource record has been maintained. So in biosharing we allow people who work on a standard or database or data policy to actually claim the record for that resource in biosharing. So that allows them to edit the record and also to see if there are any changes to the record. If one of our curators comes along and links to a record from another resource or updates some information on that record, then the maintainer will receive an email telling them what the change is, so they can essentially verify that change to ensure we haven't done anything incorrectly, or indeed learn when a database is now using a particular standard, for example. And then underneath there we have a timestamp telling you when the record was created and when it was last updated and by whom. And that's a useful way of seeing whether the data that you're looking at is current or not. Were you to scroll down the page, and I'm not going to go through all of this, you'll see a number of different sections where we capture information on the support that's available for that resource, whether there's a contact, whether there's any tools or licensing information. But what I wanted to highlight just over here are the related standards and databases. So here you can click through and browse through all of the different relationships from that record. I just want to highlight in the support section the addition of a new training link that we're adding right now, linking to the ELIXIR test portal, which captures information on training and events across the life sciences. So here you can see an example. We have the metabolites database quick tour document, and down the bottom here you can see there's a link to the metabolites record on biosharing, and vice versa. On the record on biosharing you can see a link back to TESS. Now that's the basic link that we have now, but we're working through exposing our API with TESS to allow them to also show a little bit more information about each resource on TESS, including a lot of our general information section at the top of our records, actually on the TESS website, so that should provide a little bit more information. Back to biosharing, where to scroll down the page, you can see a little bit more information, but I just wanted to highlight the credit section, where again you can see the maintainer and click through and contact them should you wish, but also who's responsible for the resource, what funding's been involved, all the way down to, if the information's available, the grant numbers involved. So that's the kind of a quick overview of the type of information that we capture for each resource. So I mentioned earlier about collections and recommendations. I'm just going to briefly touch upon those. So you can see here they're a way of grouping information, grouping records. So collections group together one or more types of resource, be it a standard or a database, by domain, project or organization. I've got some examples here. There's the CDISC standards on clinical research. We have a collection on those, which is maintained by CDISC. The combined collection, which I mentioned earlier, and then a collection here for the Australian EMBL bioinformatics resource. And then we have a number of collections, and then we have our recommendations, which are a core set of resources which have been recommended or endorsed by a data policy, more often than not from a journal or funder. And we have some examples here: Embed Press, F1000 and PLOS, and each of these we're working with the data policy creators themselves to represent that data policy in biosharing. And again, all of these collections and recommendations are curated by biosharing in conjunction with the resources involved. I'll give you an example to tell you what I'm talking about. Taking PLOS, you can see this is the PLOS recommendation. At the top we have some free text explaining what the recommendation entails, and then we have who maintains it. So here there's Emma Ganley from PLOS along with the PLOS data team. You can see a link to the policy from PLOS that created this recommendation, the homepage for PLOS, the taxonomic range and scope and data types involved. And I just want to highlight here, whenever a record has been recommended by a policy, it receives this recommended ribbon, which is shown on the record and whenever the record appears in a search list. This is quite a useful way if you have a list of resources and you're thinking, for example, which standard should I use? You can differentiate between the standards based obviously on the status, is it ready or in development or deprecated, and also on if it's been recommended or not. So that's quite a useful factor. You can see just here on the left hand side, you can refine this list by whether there's a publication associated, whether the record has been in biosharing has been claimed or not, and also if you were to scroll down you can see more information on how you can filter by country and so on and so forth. Just want to quickly show you now our graph view, which is available for all of our collections and recommendations. Clicking on there takes you a network graph representation of the recommendation, which allows you to visualize the relationships in a more kind of easily intuitive way. So here we have the PLOS policy in the center, and then linking out, this recommends all these different resources. And in gray at the back here, you can see all the other related resources which are in some way related to the resources that have been recommended. This is a good way to browse through all the different relationships. Here's another example from Embed Press this time, where you can see we also capture the relationships where a database implements a particular standard or is related to another standard, so you can see the relationships more clearly. And then finally, you can use this to reevaluate data policies, or indeed what standards you should be using if you want to put your data in a particular database. So here I've just pulled together, again from the PLOS recommendation, a number of model organism databases, and they all use the gene ontology, but PLOS doesn't recommend the use of the gene ontology. So here PLOS can, when they come to review their data policy, see this information and go, well, we're recommending all these databases that all use the gene ontology. Perhaps we should recommend the use of the gene ontology. And indeed, I have a quote here from F1000 where they've said that using this interactive browser, they can regularly monitor and update their data policies based on these new relationships that we're adding every day. Okay, so that was a very whistle-stop tour, very quick tour through biosharing. I just want to move on now to talk about some of the people that we work with, because we're very much community driven, and we work with a number of people, as you can see. I won't go through all of these. Just want to highlight some of the societies that we're working with. We work with the International Society for Biocuration, also with FAIRDom. We work with a number of research data management services, such as those from Stanford and Cambridge and Oxford, and also the DMP tool. We have a number of cross-links and data exchanges that we're trying to do to basically spread the data a little bit wider and ensure that whichever portal you're using, you're getting the most up-to-date data you can. I've taken you through the example with tests, but we're also working with identifiers.org to share some of our information on databases with them, and also with the Japanese National Bioscience Database Center to share some of our database information with the Japanese, and also take some of their more Asia-focused database information and import it into biosharing. We work with and are embedded in a number of projects and programs. I've mentioned CEDA, but we're also heavily involved in bioschemas. We work with a number of journal publishers and, of course, some standard developing groups, building recommendations and collections with them. I want to highlight now just the Research Data Alliance, as we have a working group in the RDA that is currently coming to the end of its 18-month lifespan, and as part of that we've generated a series of recommendations on how one can map data repository standards and data policies, and we've just published this for community review. You can see here some of the people who have adopted it, and you can see, if I click here, the DOI for this recommendation. As I say, it's available for community review, so if you are interested and can spare 5 minutes, please do take a look and add a comment about the recommendation, because the more comments we have, the more we can improve it and make it as useful as possible. So to finish off, I'll take us back to the start with the acknowledgement slides and thank all the people involved in biosharing and our funders, and also quickly highlight our amazing advisory board, which give us plenty of advice and keep us on the straight and narrow. And now I'd like to thank you for listening, and before I take any questions, I just want to highlight our contact email address in case you have any questions later on. Please do note that down and get in touch with us if you have any questions. And if you work on any standards or databases and you're not sure if they're in biosharing, do have a look, and if they are there and you haven't claimed them, do claim them, and then we can work together to make sure the data about your resource is as accurate as possible. Thank you.